Alright, so now we're going to move on to meiosis. I don't have a title for it, but I'm about to flip the camera and I'm going to show this drawings that I drew. They are a lot smaller than the last one, and that's because I didn't realize how long it took to make it that big, and I kind of knew before, but it didn't really dawn on me. But meiosis is basically double that of mitosis. So there's going to be a lot of drawing, so I just did it how I usually write, and I just crunched everything together. So we're going to take a look real quick. Okay, so I'll go ahead and get started. So we can start here with interface. As you can see, it's a cell. Inside the nuclear envelope, you just see a whole bunch of just DNA just put together then right there. And then we condense it here in prophase 1 into chromosomes, which, as you can see, there's crossing over here already in prophase 1, which I'll get to in a little bit later, which allows the chromosomes to kind of switch up genetic information within themselves, which that where it connects from the purple, or it kind of, I guess it looks kind of black, but it's purple and orange. That's the chiasmata. I know I butchered that, but that's what that is. It's just it's the physical link between two chromatids that like come from different chromosomes, so to say, that are cross-contaminated, I guess. Okay, so from after prophase 1, we'll move over to metaphase 1. And as you can see, I meant to put these a little bit closer together, but I couldn't really put it together. But they're side by side. That's when it come in as the synaptomal complex, which it's a protein that just mediates recombination, which allows them to kind of switch information and so on. But back to metaphase 1, it's just like in mitosis. They align kind of symmetrically in between the two bipolar spindles. And once we move over here to anaphase 1, you can see that they're pulled away towards their respective spindles. Now I did kind of mess up here, so we'll jump over here to telophase 1, which likewise to mitosis is the beginning of the split. You can start seeing the nuclear envelope develop once again, and I can kind of zoom in a little bit. You can see that these chromosomes are already different from the very beginning over here. Oh, no, not that one, over here. And that's due to the crossing over. It's not going to be the same, unlike mitosis, where mitosis, they would all be the same throughout the entire cycle. And so after telophase, we'll move to cytokinesis 1, which they completely split after that cleavage furrow and all that happens. They'll become two daughter cells. They still have two chromosomes in each cell, but there's a difference between meiosis and mitosis. Meiosis has a whole nother cycle, whereas mitosis would have been done and it would have been identical instead of these different chromosomes. So we move on to prophase 2, which is just like prophase 1, except now we have fewer chromosomes, since we're dividing it up into more daughter cells. We move over here to metaphase. They line up again. They align in the middle of the spindles, so they could be kind of pulled apart over here in anaphase 2. And as you can see, this is all kind of the same. It's just a repetition of its first go around, except with half as many chromosomes to begin with. So when we move over here to telophase, and they start splitting off into their respective cells, you can see that the chromosomes are even cut in half once again. So once we move over to cytokinesis 2, you'll be able to see that we have 4 daughter cells that are haploid. whereas mitosis, here, I'll flip this over so I can kind of talk a little bit more. Hopefully I aligned it right. Okay, so as you can see, there are a lot of many similarities in mitosis and meiosis. There are a few differences, major differences. Like I said, the outcome is completely different. So here in meiosis, we have 4 daughter cells. Like I said, they're haploid, so they have half as many chromosomes than a mitosis daughter cell, which there's only 2 daughter cells. So they have double. Sorry, I know I'm just repeating myself. They just have double the chromosomes compared to meiosis. Now, mitosis just one time around. There's no prophase 2 or anaphase 2, unlike meiosis, which they go twice. Now, meiosis does allow for crossing over, which allows for different. It allows for genetic variation, which is good for evolution and just being able to have changes in DNA. Whereas mitosis, it would be identical throughout the entire process. So the parent cell would be exactly the same as both the daughter cells. Whereas the parent cell here, meiosis, and all the daughter cells, all 4 of them, none of them would be the same because of crossing over that occurs in prophase 1. Now, I wasn't at the webcam for class due to work on last Thursday, so I don't know the exact 2 events that you were talking about that you wanted us to speak of. But not only is crossing over one of those events that does help with variation, genetic variation, but also independent assortment, which always happens before crossing over, and it can never happen afterwards. And it's just a random combination of the chromosomes, which would also happen in prophase 1. But I'm thinking, I don't know if that's what you were meaning to or not. I should have emailed you, but I just completely dropped the ball on it. But besides that, a couple other differences is meiosis is mainly for germ cells, whereas mitosis is for the somatic cells. And let's see what else. And yeah, those are kind of the major differences that I jotted down between these two.